So I think it's interesting that this is being considered a natural alternative when it functions seemingly identically to chemo.
This is an interesting topic because fenbendazole is actually used for animals. So it is not FDA approved for human use.
That's really critical too, because we can see anti-tumor effects for a lot of compounds. But the challenge is always, is it attacking the tumor or the host more?
There is this individual who had small cell lung cancer, and essentially he was given a couple months to live. He also took fenbendazole, and he very clearly states that nearly every single person on the trial passed away around him, and he was the only one left.
Welcome to Cracking Cancer, where we tackle the tough questions about cancer with real patients and world experts. Our mission is to empower the cancer community through education, bring transparency to the oncology system, and drive funding for critical cancer research. Whether you're facing a diagnosis, supporting a loved one, or simply here to learn, we're here to bring clarity, hope, and action to the fight against cancer. Hi, everyone. Welcome to Cracking Cancer. I am your host, Dr. Kyle Concannon. I'm a physician scientist at the University of Colorado, and today we are talking about fenbendazole. And joining me is Rob Swanda and Samantha Morrell. Perhaps you two can kick off your intros and we'll get into it.
Hey everyone, Samantha Morrell, patient advocate. Happy to be here. Thanks, Kyle.
Hey everyone, Rob Swanda here, science communicator, helping people to understand complex science really simply. Great to be back.
No, thanks for joining again. And we recently did our Ivermectin episode, which was a big hit, and we wanted to kind of continue that momentum in looking a little bit into the scientific literature to figure out what is fact, what is myth about some of these compounds that people have a lot of hope in. And we've done the same thing here with fenbendazole, a deep dive into some of the scientific literature in hopes of shedding some light as to where promise may be, where promise probably isn't yet. But before we kick off there, Samantha, I'd love to hear your perspective as a patient, not only what you're hearing in social media about fenbendazole, but your personal experience navigating the fenbendazole world. What do you think?
AKA Fenben, because I can't say the full word. So I tried Ivermectin for about 6 weeks. Fenbendazole was a little bit of a different story. I actually tried that one immediately as I got diagnosed. And I took it for about 8 months, 7-8 months. Again, through the traditional chemotherapy. I didn't want to take it during my targeted therapy in case there were interactions. But I thought, what's the harm, right? The reason why I was so intrigued by it was there is something called the Joe Tippens protocol. And there is this individual who had small cell lung cancer. And essentially he was given a couple months to live. And they put him on a trial at MD Anderson that happened to be Keytruda at the time. But he also took fenbendazole. And he very clearly states that nearly every single person on the trial passed away around him, and he was the only one left. And so he credits fenbendazole as the differentiator that cured him from small cell lung cancer. And, really piqued my interest. Back in the day when I was first diagnosed, I didn't know as much as I know now. So I was like, I'm going to try it. It's a lot easier to get than Ivermectin. I'm not going to go into it, but it is. So I tried it, and I didn't have any side effects from it that I'm aware of, but that's just me as an individual. And I took it along my chemotherapy journey, essentially. Ivermectin and fenbendazole are very much like put together. They're grouped together all the time. Like if someone comments, have you tried Ivermectin? It's always, have you tried Ivermectin and fenbendazole? They're never separated that I've seen. So again, they're kind of lumped together, but I think they need to be separated and discussed differently because they are pretty different compounds.
Sure. Thanks for going into that. And are you able to share the dose, the route of administration? Were these pills? Was it an IV? Like, what did you do?
Yeah, it was pills essentially that I bought online. It was a bioavailable version because fenbendazole needs to be taken with fat. So it had a fat substance in it. Again, I don't want to go into the dosage because I don't want people to necessarily be swayed by this. And I don't want to say where I got it from, but yeah, it wasn't cheap. And it was something that I thought, again, like, what's the harm? Because I was relatively healthy other than having stage 4 lung cancer. So I felt like my body could handle it. And as I mentioned, there's a whole protocol around it. A lot of people swear by it, the specific protocol. It's not just fenbendazole. It's a list of vitamins that he took as like a recipe, so to speak, of his protocol. And people follow it pretty religiously, and they say that it helped them. Now, I think the curative word is a very far stretch. Did I ever think it was going to cure me? Absolutely not. But did I think it could help? Sure. Maybe.
And when you were figuring out where to get it, I don't need to hear like website details or anything, but did the places you were looking seem credible? Were you confident that what you were buying was the real thing?
Absolutely. There are some very sketchy things out there and very sketchy websites. The one I happened to get it from was actually from an individual who claimed that they took fenbendazole and cured their cancer, and they wanted to essentially spread the word. And so they created a website manufacturing of it and made it available to anyone who wanted to buy it. But again, what a good revenue source for those people too. So, I don't know.
And before we jump into the science, can you speak a little bit to the social pressure that you were feeling to try fenbendazole? Like, what was that like interacting with your friends on social media and the inputs you were getting?
Yeah, I mean, at the time, I wasn't super out there on Instagram about my diagnosis and stuff, and I wasn't getting a ton of like, you need to try this. It was more of me doing research and just coming across this protocol because, again, the guy had lung cancer. While it be a very different type, it was lung cancer. So it very much piqued my interest. It comes up a lot when you do research. It's not like it's hiding in the internet. It's very much there. But I will say I definitely felt more pressure trying ivermectin, which is why I tried it for that 6-week period when I thought things might be not going so well anymore, because I did feel that innate pressure to try. Fenbendazole for me was more like I just did the research and I wanted to see what happened. During traditional chemo, I thought my personal opinion was, what's the harm in trying all these different vitamins and seeing what happens? And so that was really my experimental phase. And I did end up getting an integrative oncologist who actually helped me a lot formalize my plan. And he was also a proponent of fenbendazole and me staying on it. So that's why I kind of stayed the course with it.
And was your other oncology team who was prescribing the chemo, were they aware?
No, absolutely not. No.
All right. Well, as a medical oncologist myself, I'll bite my tongue. Well, thanks for sharing. I know this is it's a vulnerable space.
Yeah, absolutely.
And there's no shortage of negative sentiments from one camp or the other around here. So I think whenever you put yourself out there in this area, you're really putting yourself out there. So thanks for doing that.
Yep.
I appreciate that's vulnerable. Rob, I'd like to switch to you as the scientific liaison or medical communicator here. What is fenbendazole?
This is an interesting topic because fenbendazole is actually used for animals. So it is not FDA approved for human use. It's used in animals to help treat parasitic infections. And the way that it works is that when this drug is consumed and it makes its way to the parasites, it's going to be absorbed, and then there are these tubules inside of the parasitic cells that are called beta microtubules that help to allow the parasite to move. It allows it to move nutrients around within the cell, but those individual beta microtubules. You can almost think of like a string, but they need to go through a process called polymerization, which is where they can bind together to help make them stronger. Almost think of it like you're braiding those strings together to make an even stronger rope. So you can start, pulling those nutrients around. You can help that cell move to where it needs to go. But when fenbendazole is used, it actually prevents that polymerization from happening. So those beta microtubules now can't come together. They can't braid those strings together. And essentially, they have a really hard time moving nutrients around, and it has a really hard time with motility. So that's when you can begin to see inhibitions with glucose uptake and eventually cell death and then ultimately parasitic death.
So this is targeting a system that's in the parasite, but these systems also exist in our cells too. Can you speak a little bit to the difference?
Yes, so there are microtubules throughout our cells and throughout animal cells as well. But what makes fenbendazole unique is that because it's targeting parasitic microtubules, it's actually helping to attach to certain proteins that are found on the outside of those microtubules. So it can attach to different microtubules like those found in animals and those found in parasites, but it has a higher chance or a higher affinity of attaching to the parasitic microtubules than it does to those animals. And that's what helps keep those animals safe when they're consuming it in order for it to target the parasite more so than the animals themselves.
So like maybe a way to think about that also that I've used for my own logic for these kinds of things is like 10 molecules of fenbendazole will bind to the parasite for every one that binds to the person. Is that kind of the logic?
Yes, I would say that's true because has a higher chance of going, say, to the left than it does going to the right. But that doesn't mean that if it starts getting backed up on the left, one might bounce over and still go to the right.
Okay. And is that the mechanism, the binding of microtubules, that is felt to be effective in cancer?
Yeah, so going through some of the literature, it appears that that's the suggestion, is that because it can bind to those microtubules, it can help to lead to something known as cell cycle arrest, or then the cell is not able to actually continue to divide and grow, which is critical for tumors, critical for cancer cells, and they actually grow at a faster rate than our typical cells, right? So if we can slow that down, or if we can maybe stop it completely, then the hypothesis would be that we could slow down the cancer growth or stop the cancer growth by targeting that very specific pathway. But then there's also some additional effects that it could have. Like I mentioned before, it can also inhibit nutrients being moved around between the cells. So sometimes in the studies, you see them doing glucose assays or seeing how much glucose is being absorbed, because that is also needed for our cells to use as energy to grow and divide. So that's another effect that could be used in the cancer space as well.
Yeah, because when you're mentioning this is working by cell cycle arrest or stopping dividing cells that are dividing too quickly, you're describing chemotherapy, is really what you're describing. Is it, Samantha, I'll put it back to you, when people are talking about fenbendazole and how it's used, is a mechanism usually floated out there? Like, what are people saying?
No, I would say people are saying it's a natural way to essentially clear out their cancer and cure them. I don't think people see it as or compare it to chemotherapy by any means, because a lot of people see chemotherapy as a bad thing. It damages every cell. It's not that great for you. It can make you really sick. I don't think people see fenbendazole as a comparable thing because it doesn't make people sick in the way that chemotherapy does. So it's very much like not put in the same bucket.
Is it natural?
I don't think so.
Okay. It's because here, I'm going to read out the name. It's methyl N6 phenylsulfonyl 1H benzimidazole 2-eel carbamate, also known as a natural compound. So I mean, that's just the structural nomenclature. Mm-hmm. But the irony here is, I think, and Rob, maybe you can fact check us here, fenbendazole is probably synthetic.
Yep, it is synthetic. Yep.
but actually chemo is a naturally derived product.
Right.
So
try telling people that though, right?
Yeah.
I know.
Like we came up with many chemos because they were either secreted by a different cell as a defense mechanism. The taxane is a classic chemotherapy that also binds microtubules. So this sounds a lot like taxanes, which were originally, I believe, derived from the Pacific yew tree. So those are like literally, and they've had to do like significant forest clearing back in the day to try to get enough of the leaves to harvest taxanes for therapeutic use. Now we can make it in the lab, but back in the day, it was literally come from the plants. Versus this, quote unquote, natural thing, which is entirely laboratory derived.
Yeah, so definitely a misconception there, right? Because people say, "Oh, I'm not going to go down the normal conventional route. I'm going to go down a holistic route," right? And that holistic bubble includes fenbendazole, ivermectin. And holistic to me, I don't know, it might be just a me thing, but I see that as more natural things and approaches.
I mean, that's how it's branded.
Yeah.
But I feel like we've come to a place where if it's not being recommended, then it's holistic.
Well, that's true too.
And I'm like, well, you can go drink motor oil and that's not, that's natural. We get that out of the ground. But like, so I'm not really sure I understand. Like the distinction is we just call it holistic if like I don't think it's a good idea.
It's interesting that you bring up the Taxol part too, just because it does essentially have the same target, right, as fenbendazole, as it is targeting those microtubules. It works in a little bit of a different way as it prevents depolymerization. So those tubes that have now come together to form those strong bonds in order to move things around, move nutrients around, help the cell move and grow and divide. Now they can't move. So essentially the cell dies because they can't rearrange their microtubules, but it's having the same effect in terms of fenbendazole, whereas that downstream target is the same.
Yeah, and I've always thought of these in this context, these tubules, as really the scaffolding of the cell. Like if the cell is a building and it's all made on scaffolding, that scaffolding has to be broken down and expanded so cells can move around, they can interact with things, they can reshape. And it's also how they divide. They use that scaffolding to divide. So if you just make everything solidified the way it is, that cell's going to die. It needs that plasticity. It needs to be able to restructure itself. So I think it's interesting that this is being considered a natural alternative when it functions seemingly identically to chemo. And that's not nothing. If it does function in that way, chemo is still one of the most effective things we have. So if the premise is that fenbendazole is acting as a chemotherapeutic, well, maybe it's not how it's been initially pitched, but that doesn't mean, it might not still have some utility. So maybe with that, Rob, maybe you can tell us some of the preclinical data or some of the research that you've done into fenbendazole being used against cancer.
Let's start out with some of the early stage cell culture work. We talked about cell culture in our last episode. So just simply growing cells on a plate in a petri dish. You have to keep a lot of the conditions the same. And they're treating them now with fenbendazole or with a control. And some of the cancer cells that have been looked at include lung, colon, cervical, or glioblastomas. And what they were doing is treating these cancer cell lines with different doses of fenbendazole and then looking at growth at 24, 32, or 48 hours post-exposure. And what they began to see was that those that were treated with fenbendazole, and as the dose increased, this effect was also more seen, were growing less. So they were looking simply at growth assays. These are very easy to do. You put the same exact number of cells in one dish that you do in the other. You change only one variable, and that is one's treated with fenbendazole and one is not. And then after that time period, you go back and you count those cells again, and you look at the rate of how much those were growing. So that's kind of some of the earlier stuff that they had done in cell culture. Then they began to do some more biochemical assays looking at what we were talking about before, how it affects parasites known to stop cell cycle arrest, known to affect glucose transportation. They did some assays looking at those exact things, saw that there was certain cell cycle phases that were affected more than others. They looked at the glucose transporters as well as glucose movement within the cell. We're able to see that some of those were also affected. So this gave them a baseline of knowledge to then move out of some of the cell culture work and then begin injecting into mice. So I'll focus on one of the studies that actually showed a difference, and we can talk about that first before we talk about another study that did something similar and that did not show a difference. So we have both sides here. So they then took the cells that were lung cancer specific and injected them into immunocompromised mice or nude mice. So these are mice that don't have an immune system. They treated the mice with the fenbendazole. So the fenbendazole was present in their food. It was suspended in oil, but it was placed in the food that the mice would eat. And so not injected. And then they measured the tumor growth. And what they saw was that those that were treated or those that were consuming fenbendazole had smaller tumors that developed than those mice that were not consuming the fenbendazole. It didn't show that the tumors did not grow. But what they did not show in those studies was what the effects that this was having on the mice. So that I think this is pretty critical that we also want to look at when we are treating any organism and any animal in preclinical is what is going on with the mouse as a whole. Are they still healthy enough? And these are very small mice, right? They're just measured in grams, but typically what we need to do is weigh those mice every time they're getting a treatment or on a certain basis, so daily. And if they meet a certain threshold where the mice has lost too much weight, then the mice has to be removed from the study. So it can be similar to a clinical trial. If you begin to see someone is having adverse effects, we need to remove them from that trial. And in this study, they didn't provide any of that data. So they didn't show anything related to what this was doing to the mouse itself outside of just removing the tumors, excising those tumors, weighing the tumors, growing some of the cells in 3D culture now, not just on flat petri dishes. And although that was consistent, we didn't see those additional health implications that this could have on the organism itself.
Yeah, and I think that's really critical too, because we can see anti-tumor effects for a lot of compounds, but the challenge is always, is it attacking the tumor or the host more? And we can get at that through toxicity profiles, like how toxic is it? So in a mouse, the mouse isn't going to tell you it's feeling nauseous. It just doesn't eat. And that's how it loses weight. And that's how we surmise that this is a toxin that will probably induce nausea. It's not going to tell you it has liver toxicity. You have to look for that. I didn't see that here. And, Samantha, you can probably attest, when you were on chemo, what did it do to your counts, your white blood cell count, your red blood cell count?
I mean, I'm a weirdo, I guess. Nothing.
It had no effect at all on your counts?
No.
Yeah, that's pretty abnormal.
Yeah.
Most people, on regular chemo.
Carboplatin and pemetrexid, didn't impact anything really.
All right. Well, for most people, for us regular humans, it's gonna usually cause them to die to a certain degree, or at least not proliferate as much, not grow as much. So you'll see the people's white counts go down, or they can become anemic. This is pretty common, and it's a big side effect of the class of medication that this would fall under. So much so that in some of these articles, the comparator that they're using as something else that binds to these tubulins, these microtubules, is colchicine, which we actually use as an anti-inflammatory. And it works by binding to the microtubules within the immune system. So the fact that we're missing all that data from them, I think it is really, really important as a precaution to not overinterpret a positive signal when we see it. Rob, what do you think?
Yeah, I would agree with that. And also, it was interesting that in the cell culture models, they had a variety of different cancer cells that they were claiming this had an effect on, right? There was 5 different types. And then when they moved into the animal models, it was just one. So it's interesting because you would think that would want to be supplemented with a little more rigor. So like maybe 2 or 3 or even all 5 just to have the most amount of consistency. But we didn't see that here, which I also thought was something that just made the study fall a little bit short, because if you can show that reproducibility across cancer cell lines, that's also really indicative that maybe this is more targeting a cancer cell than it would be a healthy tissue. And we just didn't get that level of detail here.
No, I think that's another critical element. And usually when we're looking into scientific literature, when there's a piece of data that you would kind of expect to see in there and you don't see it in there, we approach that with a pretty critical eye. And I think a lot of times the default conclusion is that it was done and it's not included for a reason. And many times that's right. I think Rob, we probably both done experiments where this replicate looked weird, and if we use this model, it looked a little weird. It doesn't really continue with the story, and usually that's just not included. And that is standard for how scientific projects go. And one of the critical elements of peer review. So people who may not know, peer review is a process that when you're submitting a manuscript, when you're submitting all the research you've done, they send it out to other researchers that are in your field. And those researchers say, hey, wait a minute, you were supposed to have done this. Where is that experiment? And they're the ones that are the checks. So when someone is quoting an article that's not from a peer-reviewed journal, those checks and balances to make sure, at least to some degree, that that journal article did things appropriately is missing. Like, that's a critical element. And I will say there are still peer-reviewed journals where they get whoever they can to review it, and that takes a lot of time out of a researcher's day to review a full 20, 30-page manuscript. So even for journals that are peer-reviewed, they're not all the same caliber. And these are things that is the kind of hidden curriculum or, reading in between the lines that it's helpful to be in the field to navigate.
What do you believe are the side effects of taking too much? Like we touched on the ivermectin one, that there's a lot of potential like brain things like headaches, dizziness, things like that, as well as liver toxicity. Do we know what high doses of fenbendazole can do to us in a negative way?
Rob, did you come across anything? I can surmise.
I did not. No, I didn't. I didn't even come across anything that was related to animals that were consuming too much.
Okay.
Yeah, and the challenge with a non-FDA approved drug is you can't study that.
Okay, that's why we know more about ivermectin, because it's actually approved for parasites in people.
Yeah, I mean, me saying, hey, I put my patients on this thing that's not FDA approved, that we usually use in horses, is probably a pretty good way for me to get my license revoked. So I maybe I'm wrong there, but it's definitely, people won't be chomping at the bit to put their patients on something this unproven in humans. But, if it's working by binding to microtubules, then you would think it's going to have similar toxicity to the taxane class or colchicine, that anti-inflammatory, which again, that works similarly, and we don't use it as a chemotherapeutic. It works preferentially in the immune system, we feel. But the symptoms people can get, you can get the nausea, vomiting, you can, in the GI stuff, like anything that's a dividing cell is susceptible then.
Gotcha.
You can get your hair loss, you can get your skin reactions, you can get the taxanes can also cause neuropathy, numbness, tingling in the fingers, toes. It's hard to know because even though they're within a class, there can be differences within that class of things. So every compound is a little bit different. But at an effective dose that targets cancer, I would expect toxicity, just like with chemo.
Right.
There's nothing magical here that makes this different. Although, I will say, to give it credit, it does look that, unlike ivermectin, there is some anti-tumor activity that is probably real. It's just no one's done the job of parsing out, is it safe? And does it disproportionately target the tumor more so than what we already have? We already have taxanes. So I think that's a critical element that I want to make sure people understand, is we already have things that work this way, and they probably work this way much better. So the amount of energy to push this through additional testing just to get another taxane that probably works less well than the ones we have, that's not a really appealing ask when we have other therapies that we feel are much more appealing, like the thing, Samantha, you're on right now. Like, if I could develop that or another less good Taxol, like I'm going to choose the targeted therapy.
Yeah.
Yeah, and I was also thinking as we were reviewing these studies too about advancements to fenbendazole or other classes of drugs like this in the future, because as we mentioned in the ivermectin episode, we love when people are coming up with innovations and ideas and new pathways forward, or could we try this? Is this possible? And then diving into the science and what's actually there. So, maybe there would be a future where there could be modifications made to this molecule in order to help it be more specific to a cancer type of microtubule, or maybe it could be attached to a monoclonal antibody that could direct it to a cancer expressing cell. These are all just hypothetical, but I don't want to throw out that we're just completely against this type of thinking or this idea. It's just more so like Kyle had mentioned, it's not at the stage right now where somebody would just take this and it would go right to the cancer. You need to think about a lot of other effects that are ongoing, but it could certainly be possible, or could have some really cool thought processes and innovations moving forward.
Yeah, and I think to the credit for the individuals who are putting their hope in here, I feel bad for the individuals who were really strongly in the ivermectin camp, because I didn't see a whole lot of activity there. Like, I'm not really sure where this came from. But in fenbendazole, there's a manuscript here. Fenbendazole acts as a moderate microtubule stabilizing agent and causes cancer cell death by modulating multiple cellular pathways. It's in a peer-reviewed journal, Scientific Reports. And they went through the real process. They have some scientific rigor here. They tested real hypotheses using validated models. They're many of the appropriate questions to ask. And they, as I'm going through here, they're looking at how this molecule, we think it binds to microtubules. They do the experiments to see where in the cell cycle are the cells being arrested. Is it actually doing the thing that the taxanes do? And they combine it to another one, colchicine, that anti-inflammatory. I'm not sure why they don't also include the taxanes, the things we actually use to treat cancer. So that's another kind of confusion of mine. But they at least go through the process, and they show that, you do see more arrest in G1. And then they actually do the experiment where they do the dose curve. And this is the one that, the proof is in the pudding. How are the cells actually responding to the drug? So they take here two cell lines, H460 and A549s, which are two non-small cell lung cancer lines. They're driven by KRAS mutations. And they do that experiment where you plate them out in a dish and you treat them with escalating doses of drug. And you can look here at, you start to get some activity at about 200 nanomolar, which in reference, that's real. That's a real activity. It's nowhere close to as sensitive or as active as what you're on, Samantha. It's still about 10 to 100-fold less potent than a tyrosine kinase inhibitor, but it's in the realm of chemo. And by the time you get to 1 micromolar, you're at about half the cells are dying. And that's about the dose that we'll see around chemo, give or take a little bit. So to me, when I look at that dose, I'm saying, okay, well, that's pretty reasonable. That actually is an anti-tumor effect at a dose that seems not crazy to me. Earlier, we were talking 10x the dose of ivermectin. That's where you're, and you have to have some appreciation of these studies and have done the experiments, I think, a little bit to get a sense for just how different that is. But, at sub-micromolar doses, we're in the nanomolar range, you're starting to see activity. Okay, well, maybe it's not great, but it's something. So I think the claim that there's anti-tumor activity here is real according to the data that they put forward. Now, I haven't seen this recapitulated. Maybe it's out there. But the other thing they did, which I think was interesting, is they looked at P53 status. So they have some cell lines that have normal P53. For those who may be new to this idea, P53 is a tumor suppressor gene. When that protein is present, it can sense cell damage and then really have the cell undergo a self-killing. So it's kind of a way to protect the organism, the body, from these cancerous cells or from abnormalities. The cell will digest itself. So when P53 is present, that's a good thing. It can essentially help tumors self-correct and eliminate themselves. But it's also the most commonly mutated protein in cancer. So most cancers do not have active P53, or at least not normal P53. what they show here is if P53 is active, fenbendazole doesn't do anything. You have to have a loss of the P53 system, which on one hand tells me, well, then it's probably more likely to be real. When you see something that's too good to be true, it is. But here they're saying you have to have a tumor suppressor function in order for, you have to have that DNA damage sensing mechanism, and it's dependent on that. So they have one where they essentially give back P53, and then fenbendazole works in a cell line that lacked it. But originally, fenbendazole doesn't work. So to me, when I start seeing these mechanistic evaluations, then I'm starting to think, okay, you've thought about this a little critically. You've done some perturbations that give us the correct context in which something might be effective. This is when I start to think, okay, there may be some activity there. It may or may not be worth it, but at least the biologic plausibility ends up being present.
Kyle, can we unpack that in layman terms a little bit more? So that was even over my head a little bit. So as part of my mutation makeup, I have HER2, but I also have TP53, right? So which is very common, like you said, with many cancers. Are you saying that the study says if you have TP53, there is evidence it works, or if you don't have TP53, there's evidence it works?
If TP53 is normal, not mutated,
okay,
then it may work.
Okay. So in my case, where it's not normal, which is for most people, that, right. Okay, that means that there isn't proof that it works. In
this very limited data set.
Yes, understood. Okay.
These are a couple of cell lines in one disease type.
Yep.
And this is one thing that is really hard to convey, is the amount of rigor you need, meaning like the number of perturbations you need to try out, how many different models, how many different tumor types, in order to make these broad conclusions. So what I can conclude is in the model systems they used, you had to have working P53 for this thing to work.
Okay. All right. I just wanted to clarify that because I was concerned that people that see TP53 on their reports are suddenly like,
I have it,
so that means it's going to work. But it's actually the opposite. So, okay.
The data they have suggests the opposite.
Okay.
I can't tell you the biologic truth.
Understood.
I can tell you what this data suggests in this one cell line that they did that in.
And Samantha, you mentioned TP53. And just for those who are aware, that the TP53 is the gene. So that's what you may see being mutated. Whereas the P53, as Kyle was mentioning, is the downstream protein that's actually produced, right? So it's the same thing, essentially. Okay. I don't want people to get confused. It is the same, but one is the name for the gene, one is the name for the protein. They're not two different things. Yeah. Okay. Hopefully that's helpful. But if you have a mutated gene, you're going to make a mutated protein, if that makes sense. So and then maybe that protein doesn't work, which is what we were getting at.
Yeah, and the different in naming between genes and proteins, even though it's all part of the same thing, confused me forever. But they draw this conclusion, and it's based on they had two cell lines that have mutated P53 that isn't working, and two cell lines that had intact P53 that is working. And in the two cell lines that had the intact P53 that is working, they got some benefit. In the ones that had non-working P53, there was no benefit. So they concluded, well, the biggest difference we know about is P53 status.
Gotcha.
Maybe there are other differences. Maybe that's not the only thing. They didn't go into the mechanism and things like that, at least not to a tremendous degree. But they also did this experiment here, which I applaud them for, because I'm not sure they had to do this, but they took a cell line and they added the fenbendazole. They treated with a micromolar of fenbendazole, but then they also added in healthy lung as another control, and the fenbendazole seems to have killed both. So it killed more of the tumor than the healthy lung. The healthy lung looks like about 30% died. In the tumor, looks like about 60% died, but at 72 hours. But, you can't really do with 30% of your body dying. So this suggests at least at 1 micromolar, which was about half the effective dose, the data they have suggests there's probably real toxicity there. At least that's my interpretation of their data. Because again, they didn't go into the mouse work in this study that showed, is the mouse feeling like garbage? Are they not eating? What other effects? So at least in the 2-dimensional culture, it also affected how the healthy lung cells were growing. And, I commend them for doing that. Otherwise this is an experiment where they did other good work. They planted out cell lines in gels and looked to see if colonies formed. These are called colony formation assays. When the drug was there, they didn't form as well. When the drug wasn't there, they formed just fine. So I think these kinds of experiments show that there is a biologic mechanism by which fenbendazole can inhibit tumor function. And they go so far as to say you probably need this P53 protein to be doing its job. What it doesn't answer is it at a physiologically tolerable dose? And is it any better than what we currently have? Nowhere in here do they compare it to a taxane, which is the drug we use for this. Or maybe they did, and they didn't include it. That's another very real possibility, is they show that the taxane works 10 times better, and it's better tolerated, but that's not a very good paper. This is another thing I want people listening to think about, is when you're seeing these papers, the person who's publishing it has every intention of publishing in the best journal they can. They want their data to be shown in the most impressive light. If they publish a paper that says fenbendazole works 10 times less well as paclitaxel, no one is interested in that paper. If they remove paclitaxel and they just say fenbendazole has anti-tumor activity, well, then people are interested. So this framing and a careful eye for the controls that are missing is helpful sometimes to get to the ultimate truth, because the scientists are biased. We hold them in high esteem, but they have careers. They want to get their stuff published in good journals, and that's the system we're in. And that's the benefit of peer review, and why everything needs to be recapitulated through other scientists too. But at least from the data I see here, I can conclude that fenbendazole seems to have some mild anti-tumor activity that does not indicate whether it's toxic enough. I don't know if it is tolerable, and I don't know if it's better than what we already have, because they didn't do those comparative studies. And those are necessary. Like that's not a trivial thing. There's no way that you would get a new medicine into the clinic without a control. These do not have a control. So that is very, very critical.
I think that there was one other one that was done a few years later, which had a very similar way that it operated as the one that we just talked about, in terms of it started off looking at cell lines and then moved into animal models. So I think this is another great one for us to dive into. This one was looking at a different cell line. So it was looking at lymphoma, and then it was also treating it with different doses of fenbendazole, first in cell culture. And they did see very similar differences. Cell viability or cell growth was delayed. It was decreased. There was arrest in the cell cycles. They were focused more on the G2M phase, which is just another phase. For those who are listening, there's a couple different phases of cell cycle that you can arrest at, but basically still showing that the cell is stopping its division, which is great for what we're trying to look at related to inhibiting cancer growth. But what was different here is that when they began moving into mouse models, we didn't see the same things that the first paper saw. So they also injected mice with these lymphoma cancer cell lines, mice that don't have an immune system. They're not able to attack that lymphoma. And then they fed the mice fenbendazole with their food. And what they didn't see was any survival benefit of the organism, no change in the tumor proliferation, no change in the tumor size once they sacrificed the mouse and they cut out the tumors and looked at them, weighed them, analyzed them. And what they did see that were striking differences was that those mice that were treated with fenbendazole actually had a decreased weight. So that is a critical part of the study that was here, that was missing in the other study, which could signal either one, off-target effects, or side effects from the medication, or maybe both. And that's something that we saw because the mice were losing more weight with those treated with fenbendazole. That means it could have been doing something to healthy tissue that it wasn't doing in those that were not treated with fenbendazole, of course. So that was a pretty big difference in that study that was done a couple of years later.
Oh, that's huge. Thanks for ironing that out, Rob. And one question I have is, we're talking about mouse models, and we keep bringing out this critical element that, oh, these do not have immune systems. Can you speak a little bit to why that's a thing? Like, why do they not have immune systems?
Yeah, so if you are treating a mouse with an immune system, or honestly, even your own immune system, and you inject it with a cancer cell line, your immune system wants to destroy it. It's a foreign cell that shouldn't be there. So the only way we can actually grow tumors in mice is, one, by having them have a genetic mutation, so a specific breed of mice that is able to actually create tumors on their own, which there are those varieties of mice to study certain types of cancer. Or the other way is to actually inject them with cells that are grown in culture, and then you concentrate them down, inject a certain amount of cells into the mouse, and then allow that tumor to begin growing before you start testing a variety of different drugs or different or other therapeutics against how that tumor may grow or not grow. So if the mouse has an immune system, when you inject it with those cell lines, those cancer cell lines, the immune system will go after and start destroying that cancer, so the mouse won't develop cancer. We don't want the mouse, the mouse's immune system doesn't want it to have cancer. And also those cell lines, for the most part, are typically coming from another organism. Now, most of them are derived from humans, but you can also grow other cell lines from other mouse models or other organism models in cell culture. But typically when we're injecting them into mice, they're human cells. So you're actually growing a human tumor inside of a mouse. And again, that's another signal for the immune system to step in to say, that is not even one of our own cells, so let's start destroying it. And then it can affect the results of your study when you're actually trying to look at how is this drug operating, because we're really trying to eliminate as many variables as possible. And the immune system is essentially a variable. And so in these studies, we have to eliminate it.
Yeah, I think really well said. And for those who are listening, I want to do a different episode about mouse modeling, because there are so many different types of mouse models. And if you're thinking about like the hierarchy of how representative they are for a human, this is the bottom. This is as low as it gets. This is as unrigorous. It's usually only used to see, does my thing have any anti-tumor activity at all? And then you may go into a more pure model, something that is a mouse-derived tumor cell in a mouse body, and that will have an intact immune system. And then you can even do things like, if we're studying lung cancer, we can have tumors that originate in the lung, and then they're around lung environments, and then we can study it that way. So you get tiers of how good these mouse models are and how much you should put weight in them. The mouse model like this, where you just do an immune-compromised mouse model, usually we put these tumors in the side of the mouse. That's not usually where tumors grow. It's like under the skin. The vasculature is all different and things like that. But it can give you a sense of, does your drug, one, have any tumor activity, and two, is the drug itself toxic? So that's one of the biggest elements of this kind of mouse model. The fact that they don't include the toxicity data, at least in the manuscript I was reviewing, and Rob, you're suggesting that when they do include toxicity data, it doesn't look good. That's pretty suggestive that at least at the doses that are physiologically necessary for this to target cancer, the mice probably don't tolerate it.
Yeah.
Was that your conclusion as well?
Yeah, I would agree. I think that the mice were given essentially a similar dose, if not more, than what is typically given for a larger animal, such as a dog or a horse that is on this medication to treat parasites. And so that's a higher dose for a lot smaller of an organism can really cause some additional effects, which I think is why it was important that we looked at that paper that did see that decrease in body weight over a pretty short period of time. I mean, we're talking about a mouse that only weighs a few grams. If they drop below 20% in just 14 days, that's a big change.
Yeah, that's pretty toxic.
I mean, if we lost 20% of our body weight in 2 weeks, that would be concerning.
Yeah, yeah. Although now people are going to be buying fenbendazole for a different reason. No, and it's interesting. You know, I would say these kinds of elements where you find, is there any activity at all? While I'm not concluding that this is ready for prime time, it's also not in the bucket that this is worthless. So I think there could be a world where maybe this is combined with something. Maybe if someone really does do the more rigorous mouse model testing and can conclude that this is or is not toxic in different circumstances, like I don't see the conclusive evidence that this is an absolute throwaway in every context. But the signals we're getting are nowhere close to as good as the stuff we get with other things. There are tons of therapies that are coming out all the time where you can, double the effect that we're seeing in these, way more effective and no signs of toxicity. To be able to cure a mouse is not as high of a bar as people think it is. We do this all the time. And even the mouse model that they used here, they implanted some cells in the flank, and it seems like they immediately treated with ivermectin before even a real tumor formed. And then they just measured, does a tumor form eventually or not? And that's a pretty weak system. That's not taking a big bulky tumor and trying to shrink it down. That's taking, 1,000 cells and seeing, can we prevent them from growing? So I think the models we're seeing here are weak models, and they're probably weak models for a reason. I'm not impressed with the data. But I would also say, to throw a bone to those who looked at this and said, wait a minute, why are you discounting this? I think there's a grain of truth to that I think the scientific community is really eager to throw away the valid conclusions that people have seen. And, my hope is that we get to a place where we can have more open dialogue, kind of like we're having right now, and hear each other out a little bit better. Samantha, I'm curious your thoughts about how do you think people will interpret the conversation we've just had? How do you interpret the conversation we've just had?
I don't know. It piques my interest. But I will say for me specifically, I take a targeted therapy drug. I wouldn't want anything to ever interact with that. I think there's a great concern there when you start adding in additional things. Could it inhibit the effectiveness of the drug that's actually working for you? So there's definitely those flags for me, and there should be those flags for other people as well. It's hard because it's like, we talk about this all the time, how expensive setting up trials are and research and everything. And so it's like, are we actually going to find people that are going to dedicate the money and time towards further developing this research to see if there's a great foundation there or not? And I'm not sure that's ever going to happen, which is kind of scary because it means people will continue to interpret or dabble themselves when it might not actually be the best path forward for them.
Yeah, and I think what concerns me about all that is that people just think we're blowing them off, and because fenbendazole has been lumped in with ivermectin and the other holistic medicines, to use your term, that, conventional medicine doesn't want you to know about, that we just blow it off. And I don't think that's really the case. I think what we do is we look at data like these and we say, okay, there's a hint of something, but like we have a vast landscape of literature, which is way more promising than this. Like, it's just not worth the time. I think that's where we fall, or that's where I fall. And I can see how it's hard when someone is being told that this is really effective. They stumble upon manuscripts like these, which do show some, they even say in the title, moderate, which is something I'm sure the reviewers made them do. That's not a resounding endorsement of their own work. So, I can see how someone is looking at this from a lay perspective and is like, but the data's right here. It's showing that it works. but you need a little more scientific background and experience and exposure to these things to know where to place it, to know how much weight to give that. And the challenge is the weight I give this based on what I'm seeing, based on the mouse data, is really low. Like we see some activity. There's also a lot of toxicity. It works like taxanes, like it's just not it's not as promising as the stuff that, my lab's working on CAR T cells, which have demonstrated potential cures in certain types of cancers. And it's like, yeah, let's put our eggs in that basket. At least that's how I feel.
Yeah, and I think another problem is that, yes, there'll be some people that sadly decide not to do conventional medicine and just try fenbendazole and ivermectin. But there are the bigger majority, I would say, that do conventional medicine, traditional medicine, whatever you want to call it, plus these other drugs. And then people accredit it to being fenbendazole, ivermectin, when really it's the conventional traditional therapy that is really doing the heavy lifting and doing what it's supposed to do. But people, say if they had an incredible response, maybe they didn't really understand or have spoken to other people who have taken that same drug and had a response like them, they might accredit it to something else. And so that obviously gets spread around. People become passionate that is the reason why they responded the way they did versus other people. And then that gets some traction. I think that's where it gets really dangerous.
Yeah. And, I forget who is the individual who has a protocol.
Joe Tippens.
Joe Tippens. I keep thinking of something Poppins, but it's Joe Tippens. But I think that's from your British backgrounds are making me think.
Probably.
Very Poppins. But no, the other confounders there is when someone has a neuroendocrine tumor like small cell, was it actually a low-grade neuroendocrine tumor, which are usually cured? There are also subsets of small cell where we do actually see really long-term good responses, potentially even cures. Did they have that? So there's a lot of other potential variables here.
Yep.
To your point, and just acknowledging that this individual had an unlikely outcome and they did this. Well, there's an association, but that doesn't mean causation. It doesn't mean the fenbendazole caused the response he got. It could have been something else. And that's why, we just, as a medical community, we hope people don't put a lot of their hope into these things when they don't really have the data. It's tough. It's tough.
One last thing on the research papers too is I would kind of classify the papers that we looked at as more of almost like an overview. They didn't necessarily dive into the mechanisms of what exactly is happening, and I think that that's sometimes what sets apart some of the top tier journals or some of the top tier papers that go into higher journals than those that don't. They did try and they began to talk about the P53 protein. They did some of the assays that were associated with what we know fenbendazole already works on in parasites. But I still think there's dominoes missing, is what I like to think of, is because when you think of a mechanism, it's really like this domino falls and causes this to happen. And then this causes that to happen and so on. And we didn't get that clear picture and that clear story here. Like, yes, maybe some of these things are beginning to happen. If it's binding to a microtubule, it has P53 involved somewhere. It's leading to a cell cycle arrest at one of these phases, but how and why is that happening? And are those additional targets that we could either further make a better therapeutic for, or is fenbendazole also acting on a secondary target that we're not clear of right now? I think that it needed a little bit more for me to really elucidate that mechanism fully. And then moving into the mouse models too, I think that that's also one last hurrah, as you could say, for a journal or for a study to try and get into a better journal. And I'm not putting that down because I've also done mouse models before. You have a decent story and you are trying to just get it one step further, and a mouse model showing some effect can help that story move out from, okay, this is just something that happens in cell culture. Well, what's really the relevance there? It's not really going to be that useful. in the grand scheme of things and in the publication field? Is it going to get cited that much? Is it going to be useful years from now as we're developing some type of therapeutic or as we're investigating these mechanisms? Maybe not. But when you can push it a little bit further and show some in vivo data, that can also help your study, get seen a little bit more, which is, I think, maybe why they did it and why it has gotten a bit more attention.
Yeah, and I think we echo a lot. You have to see it in a mouse model for people to hang their hat on it to any degree. And it's tough because there's a huge spectrum of mouse models. So that in itself is not sufficient. Maybe necessary, but it's not sufficient. And that's really tough. But yeah, I mean, I'd say that the takeaway here is that this seems like it probably has some mild anti-tumor activity, but we don't know how it compares to what we already have, including the chemos. And it seems to work like a chemotherapeutic. Not a magical pill here. This is a mechanism we know well. And it just doesn't seem to be, from what I can see, as safe or effective as maybe the taxanes or other classes of chemos that we have. But, does it mean the door is entirely closed? Maybe not. Maybe we'll find a context in which this works particularly well. Maybe there's a combination in which it works particularly well. If people have anecdotes of that, send them our way. We're happy to think about it more rigorously. But with the data we have in hand, hard to recommend this for someone, really. Yeah, that's tough. All right. Rob, Samantha, any last sentiments before we peel off here?
All good on my end.
All right. All has been said. Okay, everyone. Thanks for listening. This is Cracking Cancer. You can go to crackingcancer.org for more information. Thank you for joining us on this episode of Cracking Cancer. If you found this helpful, please like, subscribe, leave us a review, and visit crackingcancer.org for more information. Every dollar we earn goes directly to furthering the podcast and supporting vital cancer research. Because together, we can crack cancer.